Yes, Betty, how are you today?
Fine, Tisha.
Wow, that's very good.
Morning,
Uh,
Tisha.
Good morning to you.
Mm.
Jerry Piroth, how are you today?
Yes, I'm fine.
That's great. Are you ready for biology?
I'm ready.
That's good. What's good over there where you stay?
Tisha, it has rained since forever, I think. Even now it's still raining.
It's still raining.
Yeah, it's drizzling.
Wow, those are God's blessings.
No, it's cold here.
Yeah, God is blessing us. Yeah. At That's raining. It's raining. That's good. Meaning those people who are like us who stay in the dusty villages, we are relieved of the dust. But again, the mud comes in.
There's no mud in Amgongo.
I'm talking about us who live this side where there is mud and dust. Mm. We are good enough for the plants. The plants, once it rains, the rain droplets wash off the dust from the stomata, so it will their productivity.
Yeah.
Yeah, Jerry Piroth.
Yes, sir.
Yeah, what is good over that side?
Yeah, it is okay. The weather is okay.
It's good. Is it raining as well?
It is not. It stopped.
Stopped raining.
Yeah, in the morning.
Is that West Nile?
Ah, I'm this side
Yes.
of Kampala.
Oh, you.
I'm this side of Kampala.
That's good. That's good. That's very, very good. Let me admit these people in the waiting room. Bakita, Bakita,
Yes, teacher.
you are here now.
Yes.
That's very important. What's good over there?
There is nothing good.
It's just coldness, like the way Betty is also feeling cold in Namugongo.
It's shining today.
Yeah, yeah, and I told Betty that those are God's blessings, and she wanted to argue. Eh? She wanted to argue. By the way, members, I gave a cake on over the weekend, and people are not eating the cake.
What's the cake about, Tisha?
I gave a certain cake in the group. Didn't you
A
see it?
cake? A cake?
Yes, a biology cake. Yes, biology weekend cake.
Hmm, Tisha.
Did you see
For us,
it?
we don't want to eat too much sugar.
Ashed.
You don't.
We don't want to eat too much sugar.
You want to eat less sugar.
Yeah, you're on a diet.
But still on the diet that you are on, in case you feed on cassava, it will be broken down to release much sugar. Eh?
Not Tisha.
So it will not be digested.
But this one will be excess sugar.
Will be excess.
Yeah.
Yeah, I gave a cake last time. Maybe we just need to visit the group again. I gave it. It was on Sunday, I think. It was on Saturday.
Okay, teacher.
Yeah, it was on Saturday after attending a wedding. I also prepared for you a cake. The group you'll have to eat. Okay, you just get some time and eat the cake. Nabuzare, how are you there?
Teacher, I'm
Catherine,
fine. Yes, teacher,
wow, wow, that's wonderful. If you are fine,
I'm fine. And you guys?
Yeah,
we are
fine.
fine.
Okay. So, what are we discussing today?
We were talking about a cake. Did you see the cake?
Teacher, I've not yet completed the cake.
But it was for the weekend. It will expire, and once food expires, it ceases to be used.
This cake had some good yeast, so it takes long to expire.
Oh, it had some good preservatives, food additives.
Yeah, probably.
Let me hope so.
But we are going
Catherine,
to submit, don't worry, teacher.
when
Yes,
is that? Because I wanted to look at how you people are presenting your responses.
by the end of today.
Okay, let me wait. For
But
us teachers,
teacher,
we are all yes,
Or tomorrow morning.
you have to answer the cake.
yes.
I know, teacher.
Oh,
Oh,
which cake
Petra,
were you?
help me. Petra, help me
Yes.
here. Cake.
Yes?
Where is Petronella? Petronella,
Yes, teacher.
how are you? Unmute. Okay. How are you, Petronella? First unmute. They muted you accidentally.
I'm fine, teacher.
Petronella, are you okay?
Yes, teacher.
Let me hope you are not attending the lesson from a tank.
From a what?
Since your name has some component of petrol, you might go into a petrol tank and you attend the lesson.
No. I'm at home.
Okay, that's very good. That's very
Mm.
good. Wow, we need to
Okay.
start. We have someone who wants to lead us through the opening prayer. Kathy, are you the one?
Okay.
All right.
Let's humble ourselves for prayer. In the name of the Father, and of the Son, and of the Holy Spirit. Amen. Heavenly Father, we want to say thank you for this day. We want to say thank you for our teachers. We want to say thank you for our lives. Thank you that we are going to discuss this morning biology, and we call upon your Holy Spirit to guide us throughout this session. Provide knowledge, wisdom, and understanding so that we may be able to understand and remember each and everything we are going to be taught. We rebuke the spirits of forgetfulness. In the mighty name of Jesus, we have prayed and believed.
Amen.
Amen.
That's wonderful.
Amen.
Yeah. That prayer has invoked God. Betty, have you
Yes,
seen
teacher.
Kathy has invoked God?
I see. I've seen.
Wow. And this lesson is now going to be having the most stable internet because Kathy has invoked God. And I do pray that power stays on because my network depends on Wi-Fi, which depends on power. But still, when
Likewise.
it goes off, we should not worry. I have a backup plan. Fine. Okay, so welcome to today, the 16th of December, 2025. I want to thank God that we still have about how many days to Christmas? 10 days of 9 days. 9 days to Christmas.
When will we
Even
stop?
yes,
When will we stop study?
we shall have a break, a Christmas break of a week.
That's the next week.
Yeah, let me confirm here with my calendar. Next week is the Christmas week, right?
Yes.
Meaning this week here, I'll talk to Mr. Sentai and we see this week, next week.
Yes, I am.
Yes, Mr. Sentai,
Sawa.
you're
Good
welcome.
morning, neighbors.
Good morning, sir.
Like the thing is more like obvious. People should go for Christmas, and as soon as this week ends, it will be Christmas mood already. So when this
Well,
week ends, and I think we plan 22nd. From 22nd onwards, they will be off, and we shall return on 5th. It will be a Monday. So I don't know how many days are there. Oh, it's 1 week, but we shall
those
turn
are
on a Monday.
those are 2 weeks, the Christmas week and the New Year week.
Akum will be home, not thinking about anything, but
Teacher.
eating. Yeah,
Village.
Mister, you can continue. So village is not home.
Then the politicians, Nabuzare, I really don't know. We shall campaign as we study.
Nabuzal is
Shall
campaigning for who?
Yeah, someone will campaign for you as you study.
Woman
Okay.
NPO.
Yourself, teacher. Who is saying yourself? Okay. They'll switch off internet nationally. Maybe during the voting period we shall see. If they vote, if they switch off for a week, then we shall be off for a week. I don't know. What do you think? Should we study during the Christmas season so that we compensate the other week, which we shall be offline or off the net? But for me, I'll go to another country and I connect internet from there. I don't know for you people. I'll vote and I run to another country, then I connect internet from there. So I'll be on Zoom. I'll just wait for you. Bakita is refusing. Amanda is just laughing. You're killing me with laughter. Sorry, we shall bury you. Rub that from my mind. Which one? I'm going to vote and move to another country immediately after voting. Of course, Christmas is Christmas, and you should not expect me to be on Zoom the way you are seeing me here. I need to also slaughter some hens, cows, and goats. Okay, that is it for now, members. You're welcome. Um, We shall do respect Christmas because most of us are Christians, and being a Christian, it means that you follow Christ's example. So that means that since you are a follower of Christ, or since you follow Christ's example, you have to celebrate his birthday as well. All right. Today I want us to first go through the online activity that we did. I went through some of your answers, and very soon I'll be sending a report to each and everyone's email detailing giving the scores that you already got from that online activity. I'll give the scores, then I submit, I release the results to your email. And so, nevertheless, I'm presenting here so that I saw people with the different answers and different approaches. I want us to harmonize the approaches here. Let me first beat these people who are making noise. Where? Them bring them over. Members, I'm back. I'm back. I'm back. I'm from fighting these people who are shouting. You don't know some people. Which people are you talking about? Maybe we shall have Christmas on Zoom. Eh? Amanda, we shall create a Zoom link for Christmas so that we celebrate from Zoom. However, Juma, Alex, that must be Gune Tracy. At least Gune, talk to us. Say hi to us because people want to hear your voice. They can't wait to hear your voice. So Amanda, Juma is not a male, is a female. She's Gune Tracy. Don't worry, is a female.
Teacher, from where?
Come again.
Juma, from where?
Who is saying something?
Me.
Unmute, Juma. Juma, this is Gune Tracy. Gune, unmute and
Guys, it's me, Tracy.
Oh. Hi, Tracy.
Hi, how are you?
Hi.
Long time.
Tracy, some money for data.
Do I look like UN?
You never know.
I don't look like UN.
okay, members,
They
we
told
shall
us at your home you use Wi-Fi. So nice, send us some money for that.
members,
I'm just paying for the Wi-Fi.
members, let's continue. We shall continue with that jazz later. I've muted everyone, so you don't feel offended. But I think you've heard her voice. She'll continue interacting with us as time goes by. So Tracy, I believe you've not done this assignment because you've just started studying with us of recent. However, I left this online assignment and people here, the majority of the individuals who have attended today, attempted the assignment and submitted it. This assignment was given after discussing about the DNA, then discussing about protein synthesis and the sub-stages that are involved in the protein synthesis, that is right from transcription up to translation. And so a 45-year-old patient, Amina, presents with progressive polyuria, which means frequent urination, polydipsia, which means excessive thirst, unintentional weight loss, and fasting hyperglycemia. Hyperglycemia means elevated or high levels of blood glucose in blood, after even fasting. Pancreatic islets histology, meaning the study of the tissues of the pancreatic cells that contain the pancreatic cells. Okay, that showed reduced functional or function of beta cell mass, meaning the amount of beta cells that were present were few and were secreting insulin. The rest were not capable of secreting insulin, meaning this person here was diabetic in simple terms. However, if you continue, they say that with no mutations in the insulin gene, clinical chemistry indicates low insulin availability and disturbed whole body glucose regulation, meaning this person's body, Amina's body, is not capable of regulating the concentration of glucose in her blood. And so you are asked to analyze the molecular basis of insulin production, meaning looking at the different processes that occur. Since insulin is a hormone and a hormone which is protein in nature, meaning they want us to describe on a molecular basis how or explain how insulin is formed right from, or how the gene of insulin is expressed. So, the cellular control of the secretion and the systemic consequences of glucose balance. So the task there, meaning this person here was diabetic. And this type of diabetes mellitus, it's called type 1 diabetes. If you didn't know, we have two types of diabetes mellitus. There's type 1 and type 2. For those people who are obese, who are too fat, they are most likely to have type 2 diabetes, and type 2 diabetes can lead to type 1 diabetes. Type 2 diabetes is a situation where someone's body, the islets of the beta cells of islets of Langerhans detect high levels of glucose in blood and secrete large amounts of insulin. However, this insulin does not have effects on the beta cell, rather on the liver cells and the muscle cells, because the liver cells and the muscle cells have receptors which have been covered by fat layers. So those people who are fat, meaning those cells are covered by fat layers, so that blocks the receptors of insulin hormone, hence no reception of insulin. So that is the type 2 diabetes. Then on the other hand, type 1 diabetes is a case where the beta cells have been destroyed or are malfunctioning. Most of them are malfunctioning, and meaning there are few or no cells that are capable of secreting insulin, meaning there are low levels of insulin in blood or no insulin at all in a person's blood. That is type 1 diabetes. So type 2 diabetes can lead to type 1 diabetes because once the receptors do not bind the insulin hormone, there will be no conversion of glucose, no stimulation of conversion of glucose to glycogen, or no stimulation of the glycolytic pathway, or a pathway that would result into breakdown of glucose into simpler molecules, or ATP, respiration of glucose in simple terms. So since there is too much insulin being secreted and no glucose is being converted to glycogen, meaning this person's body will continually detect, rather the beta cells of islets of Langerhans will continually detect the high levels of insulin in blood. And so as such, these beta cells are forced to overwork. In medical terms or in biology, if a particular organ is forced to overwork, The consequences are that part will have cells which will become malfunctional as a result of being overworked, or that organ will be inflamed. That's why you see those people who have goiter, they have a swelling at the neck. It's because the cells in the thyroid gland are overworked to secrete thyroxine hormone. So similarly, the cells of the beta cells of the islets of Langerhans are overworked, and as a result, most of the cells begin dying. And once the cells die, it means that the levels of insulin that, rather the amount of insulin that will be secreted by such glands will continue to decrease, and that would result into type 1 diabetes. So I think we can see how type 2 diabetes can result into type 1 diabetes. Back to our question. There is the activity that we did. And if I'm to go to the task here, the first task here, how is the structure of the genetic material found in the nucleus of a beta cell suited to its function? Here, I'm not going to give you answers, members, and I don't have your answers. I'm so sorry, Bakita, I don't have your answers. You should be aware that. So you'll be the ones giving us the answers, since it was an assignment which you did and researched. Just share with us briefly what you have. Then we continue. Mark Lena. Mark Lena is attending for the first time. I'm seeing you for the first time. How are you? How are you, Mark Lena? Macarena? Teacher, under what conditions or situation can we have much secretion of insulin? Once the glucose levels in blood are high or continue to remain high beyond the normal ranges or beyond the norm, the beta cells will continually be stimulated. Remember, the beta cells, for them to secrete insulin, they must be stimulated. And the stimulus here is a high concentration of glucose in blood. So the continuous high concentration of glucose in blood results into a high secretion or much secretion of insulin hormone by the beta cells of islets of Langerhans in an attempt to regulate the glucose levels in blood. Betty, why? I don't know why you are asking that question. Why? I really don't know.
Tisha,
Unmute and talk to us. Yes.
you said that you're not going to give us the answer, and at first we were also like struggling to get the answers, right? So, Tisha, you have to give us the answer. Tisha, you have heard me. Tisha,
Akumu, you have made the teacher disappear.
no, Tisha, I did not.
Members, who is the account? I think it is an Betty was asking a question and she disappeared, and I also disappeared. So we
No,
all
teacher was
disappeared.
not. I did. I did.
It didn't disappear. Are you for real?
Yes. You can ask.
Oh, so I think I'm the one who disappeared mysteriously.
Yeah.
I think my router was just refreshing and disconnected me. Let me use an internet cable here now.
But members, have you realized the teacher is in the forest? Masita.
Mr. Sentai, here you study about life very well in the forest, receiving a lot of oxygen.
Master, won't you be attacked by wild animals?
I'll adapt and get mechanisms of camouflaging.
If that is a forest near to Amanda's home, you are finished.
They told me Amanda has a neighbor who rears some wild animals.
But Mr.
Is
Sendai,
that
we
true,
don't have a
Master?
forest near our home.
Uh, that's called violence.
For
Okay,
sure,
I think
actually. Mr. Sendai, now you've disappeared.
No. You know, some people are good at conserving the environment. Now Amanda lives almost in the bush.
all
Thank you,
right.
teacher.
Okay, I think was a nice interlude.
Yeah.
Mr. Wanyonyi, I have a suggestion.
Please do, sir. Do suggest.
Akumo should stop requesting for hard things. Now the teacher has been off just because Akumo was.
Teacher. Teacher Sentai.
Yes.
To be honest, this question was very difficult. I didn't request for it. Teacher is the one who gave it to us, and he was threatening us to answer.
So the teacher is threatening.
Akumo, you are killing me.
And he
You
told
didn't
us not
request
to
for
use.
the question.
He told us not. He told us use our heads. So I tried. You can even ask these guys.
Aman, Betty, did you submit the assignment?
Yes, I was the second one even to submit.
Yeah, okay, I remember. So Betty wants to hear my answer, yet I don't have it.
Teacher, you
Betty,
have.
Mr. Wanyu,
yes,
I have a suggestion.
please suggest.
Teacher, I'm suggesting, okay, we present our answers because, teacher, you know our answers. You mark us.
Yes.
Then after that, you also give us your answers. You say that if this is a discussion, it should be interactive. So, teacher, you're also going to interact by giving us your answers.
Exactly.
I have no problem with that, members. And your suggestion is very good, and it's what I was also thinking about.
Okay, thank you, teacher.
Yeah, so who is the first one to take us through their responses?
Akum bet.
No. You guys,
Bet-
I'm not ready.
Betty.
Teacher, you go alphabetical order.
Or maybe I should get the answers of people here and I project them and we discuss.
Yeah.
Eh?
Teacher,
No,
that's better.
No, teacher,
Yes.
no. No,
teacher,
teacher.
don't.
Wow, I like that.
Teacher, no.
Mm.
Teacher, you
Okay,
said
so who's
something.
ready to take us through? What did you come up with? How is the structure of the genetic material found in the nucleus of a beta cell suited for its function? How did you interpret that task? Yes, I'm waiting for someone to take us through. Let's unmute. Let's unmute members. Oh, I'm going to select people at random here. Members, see, they
Let me
don't want.
say.
Okay, yes, Bakita. Hey, I was not seeing your hand. I'm very sorry, Bakita.
It's okay.
Yeah.
Anyway, the question was, how is the structure of the genetic material found in the nucleus of a beta cell suited for its function? So, I identified the structure. I identified the genetic material as DNA.
Wow,
So,
that's very good. Get the first marking point.
yes, Sharon, I came up with, I'm going to give you my point, teacher.
It is very fine.
So, the genetic material found in the nucleus of a beta cell has sugar phosphate backbone, which is held together by strong covalent phosphodiester bonds to provide stability.
Wow, that's very nice. I can see how you are relating the structure to the function. So providing stability. Wow, that's good.
Yes, teacher.
Strong phosphodiester bonds. That's good. Um, Next. Oh, you want someone else to give an answer?
Teacher, we are all contributing. So I've first given my first point. Now people add, then yeah.
Members, what is your next point? Makilena, you are just seeing us from Kamori. Come out of Kamori, Makilena.
Mr. Wanyonyi.
Go ahead. Who is that?
Okay.
Amanda. Yes, Amanda.
Yes, my answer is okay. Since we've identified the genetic material as DNA,
Yes.
so it has hydrogen bonds that can easily be broken to form a fork, exposing the transcribing strand, enabling the formation of mRNA.
Come again. I like such mature answers.
So it has weak hydrogen bonds that can easily be broken, forming a fork, exposing the transcribing strand, enabling the formation of mRNA.
Wow, enabling the process of
Trans-
trans. That's very good. I like. So, Beck, do you see how people have very mature answers? People have. That's why I was saying, I don't
I've
have. I don't.
seen.
I don't have an answer. Yes, Catherine, your hand is up.
Okay, teacher.
Master.
The genetic material is DNA, right? So
Yes.
it's made up of a double helix, which is compact to store lots of information in a small place.
Wonderful. It is very long and compact so that it can store information, lots of information in a confined or a small space. I like this. Wow. You must get a gift, you people. Mr. Sentai, where are the gifts? You should be giving these people gifts.
Mister.
All right. Yes, Bukenya.
It is made up of many hydrogen bonds, which contribute to its stability.
Okay. Yeah, so it's made up of hydrogen bonds. How are they? They are strong, right?
Yeah. Hence
Good.
contributing to its stability.
That's very nice. People have mature answers here. You should be going for a medical course, you people. Uh, Next person. That's very good. I like this. Uh-huh. Are there some people in the chat? Let me first go there. I see very many chats. Betty's fault, teacher, that is you. Eh? I can't understand what's going on in the chat, but let me hope Mr. Sentai is there. Okay. Who else? Members. Those ones who have not spoken. Please speak. Bukenya has not spoken. Master, Bukenya must. Hey, Bukenya's. Isn't he the one who gave us that answer? I think so.
Mr. Wanyonyi,
Yes, please, Mr. Sentai.
I would like to hear something from, not something, an introduction at least, from
Yes.
this girl. She has even, Maclena. Maclena has
Makilena.
just joined. Maclean,
Achena.
that is Acheng, Maclean.
Yes.
Jen, you see there is an icon down, which has a speaker, where is
Oh,
mute or unmute. So you can tap that icon and then you'll be unmuted and you'll be able to talk to these people.
alternatively, Makilena, when I ask you to unmute, there is a button that comes on your phone screen. The other side says unmute, then the other side says stay muted. You click on that one that says unmute. Let's go, Makilena.
If she's not on for us, we shall just take her back.
Yeah, there's a waiting room waiting for her.
Anyway, she's from far distance. They are at the borders of Congo and Uganda, West Nile. Maybe we continue.
Wow. Zoom is also wonderful. You can even attend from the border, even from another country. Okay. That's wonderful, members. I thank you so much for your contribution. Petronella, you are quiet today. Please give us an answer, another answer. Let me ask you to unmute and don't say stay muted. Yes, Petronella.
So it's DNA is long.
Move closer to your phone. We can't. You are speaking from a far distance. Move closer to your phone. If possible,
Okay.
put it in.
DNA is a very long structure.
Mm-hmm.
Just wait.
Okay, as you are thinking about it and organizing your answer, Mark has finally unmuted. We thank God you have unmuted. Please talk to us, Makilena.
So when you review the
Members, can you hear Makilena? I don't know if it's my network.
To understand, she's from the borders.
You need to decline the tweet.
We are really struggling to hear you, and we are very sorry for letting you to unmute. Please forgive us and type your.
Mr. Wanyonyi.
And type your answer in the chat. Just type your answer kindly.
Yeah, what I was trying to say is
Yes.
DNA is a relatively large molecule and can
Yes.
carry a lot of information.
So what makes it carry a lot of information?
It's being large.
It's large, okay, long, large, and it is also tightly packed or wrapped around the proteins called the histones that ensure close packaging. So just like related to what our first presenter, I think, was that our first presenter told us about that. It can be stored in a small space or confined in a small space. So what makes it possible is that it is long and the long strands are wrapped around proteins called histones to ensure proper packaging. Just like when most of us are coming back from home to school, is it coming back or going to school? Okay. If for most of us who are going for a boarding school, in case you have very many clothes and very many items that you want to carry, and you have a small bag or you have a small suitcase, in case you arrange these clothes poorly, those clothes will not fit in that suitcase. And so we must do what? Petronella, we have left the girl and we have asked her to forgive us. She's going to type in the chat. So, what was I saying? So I was saying that in case you don't arrange the clothes very well, you will not be capable of packing your clothes, or it will be very hard for you to pack for all the clothes to fit inside that suitcase or bag. But in case you arrange your clothes very well and you fold them in an organized way, everything will be just fitting perfectly. And that's what the DNA also, that's how the DNA is also arranged inside the nucleus. Then the other issue of the DNA molecule being large, remember DNA is confined to the nucleus. And let me ask you a question. What do you think would happen in case DNA leaves the nucleus under normal circumstances? In case DNA leaves the nucleus, what do you think would happen? Makilena, you want to think? Makilena, please,
Pardon?
we are. Yes, Makilena. Finally, you are. Yes. Okay. Let's hear. Members, what do you think would happen in case the DNA leaves the cell? It leaves the nucleus. Makilena, you have kids that are beating saucepans or they are beating metals and they are a bit making noise for us. So that's why we are muting you. Eh? Yeah, don't, yeah. Members, what do you think? Type in the chat in case speaking is hard.
Bakuta's hand is up.
Yes, Bakita, I had not seen it.
I'm just trying.
Please do try.
I think in case DNA leaves the nucleus, it's likely to be destroyed.
Okay.
Yes.
And therefore, since outside the nucleus, you might find some other enzymes called the nucleases that would catalyze the breakdown of the DNA polynucleotide into nucleotides, hence distorting the information stored. That's very good. I like that. So we have the answers. Members, don't undermine yourselves. You have the answers, which are even better than mine. Because for you are being taught by many teachers. For me, I was taught by one teacher. But for you, you're being taught by many teachers. So you are better. Okay, so the DNA, it being large, it makes it impossible for the molecule to go through the nucleopores and go to the cytoplasm where we find the nucleases, which might break the DNA down. So it is of an advantage. You can also highlight that of as the structure suiting it for the function. Being confined in the cell means meaning that it being large, it's confined in the cell where few or no nucleases are found. Teacher, for this lesson, I'm a bit off. Maybe it's proper. I'm a bit off. Oh, I don't know why you are off, Makilena. Maybe the network is making you off. Let me hope so. That is how the structure of the DNA is suited to its function. Maybe if I'm to get an image here, maybe some people will get some answers from the image. Just in case they don't have a surface from where they could begin thinking. We don't have a primer for thinking. So here, let's get a structure of DNA. What does it exactly have that makes it suitable to carry out its functions? The function is to store the genetic material and also provide templates for transcription or gene expression, to act as a template for gene expression. Ask a biologist. Okay. I'm interested in this image here. Hope you can see that, though this is showing us the molecular levels. Let me zoom in a bit. Okay, someone already mentioned about the phosphate group. The phosphodiester bonds is called a phosphodiester bond. Di means two, so you have two bonds joined together by phosphate group. So it's a phosphodiester bond. Then these ones that have two circles, they are called purines. These nitrogenous bases that have two circles are called purines. Then the one that has one circle is a pyrimidine. So the pyrimidines, we have thymine and which other do we see here? Cytosine. Thymine, which is pink, the pink circle. Cytosine, which is, I don't know which color is that. I'm not good at identifying colors, but that looks like what? Peach. There's a woman who told me that color. Peach color. It resembles some orange with pink, a mixture of orange and pink. I think it is peach. Girls, which color is that? Bakit, which color is that? This one of cytosine. Bakita, you are just laughing. Talk to us.
Tell me, I think it's peach.
Oh, okay. Let me hope I'm correct. Peach. Cytosine, cytosine and thymine. These are Those are called what? Pyrimidines. Then guanine and adenine. Guanine, which is blue, the blue circles, nitrogenous base, and adenine, which is the green circle or lemon green circle. Those are pyrimidine. Rather, those are purines. They have two circles. So, can someone give us any adaptation based on the structure that we are seeing there? Apart from what we have mentioned, people talked about the hydrogen bonds, people talked about phosphodiester bonds, the sugar phosphate backbone, and so on. Any
Mr.
other
Wanyonyi,
thing? Yes, please.
I think
Yes.
Acheni what was trying to one meaning that she may need the stem of the question, like the question, the
Oh,
scenario.
sorry.
Maybe after removing this DNA, you can put for her to go through so that she can continue, like she can cope up with people. She did not see the scenario.
Oh, I understand. Thank you for that. Achen, we are going to project that. Meanwhile, Nabuzale, talk to us.
Yes, teacher. The
Talk to
DNA, okay, it has complementary base pairings to allow the molecule replicate itself accurately.
Wow, to ensure accuracy during replication or transcription. That's very nice. It's very nice. Eh? I just feel that God, we should just bless you. Moses Sentai, what do you always say? God bless her entirely.
Yes,
Let's
sir.
hear. She has given a very nice. Did you hear her answer? It's very nice.
Yeah,
Ensuring.
God should bless her
Yes.
uh, with the best grades she needs. Nothing is better than that.
Exactly. Let's also hear another person who is looking for God's blessings. That is Amanda Gloria.
So the DNA molecule has nitrogenous bases.
Mm-hmm.
The DNA molecule has nitrogenous bases that the DNA has nitrogenous bases on which RNA nucleotides are added following complementary base pairing to enable formation of mRNA, which moves out of the nucleus into the cytoplasm to be used for protein synthesis.
Okay, that is very nice. It is having about 3 or 2 marking points. It's having about 2 marking points. But that's very nice. In some cases, it is related to what Catherine was saying, but that is also very nice. May God bless you as well. I think that is it for now. How about the DNA structure being anti-parallel? Can someone relate it to its function? Anti-parallel. You have the two strands running in the opposite directions. That is from the 5' end to the 3' end, then 3' end to the 5' end. Yes, can someone relate it to the function? For members who didn't study about the DNA, when you talk about 5' end and 3' end, I think it is something new to some people. Now, if you look at this structure here, DNA is a molecule that is made up of very many nucleotides. But each nucleotide has a 5-carbon sugar, which is called a pentose sugar, and its name is deoxyribose. Five, meaning it has 5 carbon atoms. And let me check whether my cursor is clear. Okay, let me pick here something, a clear button here. Let me use a pointer. Let me use a pointer. Okay, I think this is clear. If you are to focus on this one nitrogenous base, I mean one nucleotide. A nucleotide is made up of a 5-carbon sugar. This is a sugar molecule that is made up of 5 carbon atoms. This is carbon number 1. Members, my internet is misbehaving. That's why I'm jumping on and off. But Amanda Gloria's hand is up. Please talk to us as I'm planning.
Teacher, I wanted to try on that question, the use of DNA being anti-parallel.
Yes.
So, okay, I don't have a well-defined use, but okay, this is the way I understand it. So,
Teacher, all of you from Gandhi, I don't know what you're talking about. We can't hear her.
I thought the problem is just
but
Amanda, we are very sorry we can't. We are very sorry we can't hear. Okay, we are hearing you, but your voice is in a very, very, extremely, very slow motion. We are very sorry we can't hear. I don't know. Internet is misbehaving.
Teacher?
Catherine, you wanted to say something?
Yeah, I'm like, let her type in the chat because literally there is nothing we are getting from her statement.
Okay, Amanda, just pray that you type in the chat your statement as members have asked because we are not communicating.
Teacher, can you hear me now?
We have heard you partially.
What about right now?
We are now hearing you well.
Okay, so teacher, I was saying that the DNA, okay, the adaptation was it runs in an anti-parallel direction, meaning that the two strands run in opposite directions. So I was
Yes.
saying that this enables it to be synthesized in two different directions.
Okay, it allows protein synthesis to occur in different directions, eh?
Yes, because I was saying that when it is okay, when it is unwind and unzipped, the leading and lagging strand are formed, which run in different directions, whereby the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously over following Okazaki fragments. Okay, sort of.
Okay, so
Yes,
that is during replication.
yes, teacher. Okay, it enables, yes, that, teacher.
Okay, so during replication, DNA being antiparallel allows replication to occur at the same time in different directions. That's according to Amanda. That's very good. But as well, it also allows protein synthesis to occur in different directions as well, because RNA polymerase enzyme, actually those enzymes that catalyze both replication and transcription work in their unidirection. So they follow one specific direction from the 5 prime. So briefly here, the reason as to I zoomed this picture in is because I wanted us to, for those people who have not known what the 5 prime end is and what the 3 prime end is, here, where is my pointer? Pointer. Okay, here we have this one here is, this is a nucleotide having, actually this nucleotide is called adenosine. Adenosine, having the adenine nitrogenous base with a pentose sugar and a phosphate group. So on this pentose sugar, this during naming of organic compounds, this one becomes carbon number 1, which is bonded to the nitrogenous base. Then this one number 2, number 3, every corner here where there is no oxygen, there is a carbon atom. So this is number 1, number 2, number 3, number 4, and number 5. So on this number 5, where there is a bend, we see the phosphate group bonded to it. So this is called the 5 prime end. But in case, on the other side, we have an inverted nucleotide. It is inverted, meaning it is facing upside down. So for that case, still, where the nitrogenous base binds, that is carbon number 1, this is number 2, this is number 3. So that's why you see this side we have the 3 prime end, 3 prime end. Then this other side is the 5 prime end. Yeah, for those people who might not know, who might not be knowing what the 3 prime end and the 5 prime end is, I think I've tried to explain. Though sometimes this topic of DNA is complex for some people to understand, because we talk about very many molecules which cannot be seen by the naked eye. So it's like you're speaking aliens, things that cannot easily be seen by the naked eye. Okay. Do you have someone who has a question before we proceed? Anyone with a question? Silence means that no question, or people are fearing to ask questions. But if there's a lot of silence, we shall move on to the next part. But those are the answers that were required. And I believe those people who were finding challenges in attempting have learned something and they have added something into their answers. The second task, let me first project the scenario for this girl here. I'd forgotten. There is the scenario, Makilena. 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 Are you there, Makilena? Are you reading the scenario, Makilena?
I am there, teacher.
Okay, let me give you 1 minute. You read the scenario, go through the scenario. Plus, some other people have just joined us. I'm not seeing a Gune Tracy. She has disappeared. She has run away. Maybe she has gone to look for a better Wi-Fi. Meanwhile, as you're reading, let me just go and get some water for drinking.
Macklin, should we read for you? Hi.
Kathy, read.
Please do it. No wonder you guys, for me, I'm this for this lesson. So, just know.
You want to understand my English these days? Somehow I'm
Kathy, please.
You're right now, you guys.
allow me.
Oh, it's
I
okay.
hope teacher
It's
ain't
okay.
hearing this. Wait, I hope teacher ain't hearing this, but trust me, I even don't have a book.
He's hearing you is bad.
Macklin, right on
Couldn't
your
have
phone.
had you.
Guys, if you know where I am right now, I can't even access a book, apparently. I'm at my granny's. So, let me just prepare for the next lesson. I feel like leaving.
Get
It's
a
going to
summary book.
The lesson
Never
is
a teacher. Oh.
teacher, that's the secret. Girl talk.
Macklin, don't risk leaving the work for you, so just be there and follow. Do what Dita says.
By
With
the
the
way, yeah.
man, you guys know I'm just so. Do you know I'm at my granny's? People are making noise. My cousins, grandkids. When are you guys? Oh, I'm going to leave, but by next week I'll be around at home, so I'll be maybe proper. So
Next week we don't have a lesson.
next week you don't have.
Yeah.
I've just
Yes.
received the timetable this morning.
It's Xmas week.
So
Next
for this,
month.
okay. Let me first continue reading the scenario. I'm not even understanding.
Don't worry, just follow.
People never disappoint.
Allow us.
So, Cha, you are tapping jazz, eh?
We'll never disappoint you people.
How? Teacher, how?
Yeah, you can attend the Zoom lecture, Zoom lesson minus even having a kabook.
Man,
Why are
teacher, I
you?
thought you went for water, when I.
Yes?
What the?
What's wrong with water?
Teacher, you are tapping jazz.
Yeah, just know everything is being tapped. And even
Not
if I
being
don't,
tapping.
even if I don't tap, I can tap it later after the lesson. So don't worry.
Shabad manners. Oh.
Don't worry. However, okay, let's go ahead. It seems this one is trying to read, but the reading is reading her. She's reading this assignment, but the assignment is reading her. Oh, sorry,
Teacher, I
Magdalena.
can't see clearly.
But we are just praying for you that you will settle and get time to go through this assignment. And this meeting here is being recorded. So even after the meeting, you can go back to YouTube and watch these discussions that people were doing. In case you get your whatever, your notebook and what? And pen. In case you get your notebook and pen, you will go back to the YouTube, you watch the video and note down something. Yeah, as members, these videos on YouTube are very long, and in case a video is very long, it becomes too boring. But there's an option of speeding up the video. You can maybe speed it up to 2 times so that the person who is presenting speaks faster or slower. I don't know. I don't know. But you'll find the recordings on YouTube. They'll be shared on WhatsApp. Mwanje had gone to the waiting room. I don't know what he was doing in the waiting room. Maybe he's taking some tea from there. Should call us also in the waiting room. Mwanje, you should call us in the waiting room because you were just there. I think you were eating from there. And for us people, we also love eating. Anyway, that is just by the way. But let's continue. We pray that you follow up. For those who are not following, you can save some time. You go to the YouTube channel and you watch this video again in case you had not understood it in the first place. So that's the beauty of having a Zoom meeting. You can go back and review what you discussed. It's not like a class. The teacher comes in class, he speaks. If you didn't write something down and your colleagues also didn't write, that is gone. But the good thing with this one, it's being recorded. You can still go back and go through the meetings that we've been having. Let's have another person to take us through the second item, the second task, to describe the role played by different enzymes during the formation of mRNA from DNA. Before someone comes in, I think people should be raising up their hands. As I'm waiting for you to look for a hand for raising, whenever you hear a person or a task that requires you to describe the role played by either an enzyme or an organelle or a structure in a given process, it simply means that this examiner, in other words, he or she wants you to describe that particular process. However, he wants you to put a focus on these enzymes. The way you are putting, rather, the way you are phrasing your enzymes, they should be in a chronological order since it is a process. Step by step, the first enzyme should come first and the next up to the last enzyme. So in case there is a breakage of the sequence of presentation of those enzymes, then the process ceases to be fruitful, and therefore you get the answers wrong. But however, in case they ask you to describe the role played by different enzymes during the formation of mRNA from DNA, in other words, this person is just asking you to describe the process of transcription, but in your description, you should highlight the enzymes mentioned, the enzymes that are involved and their role and their function during that process. Another person would ask, describe the role played by different cell organisms. I think that question is there. We shall go and look at it. It's the same question that I was also thinking about. Maybe describe the role played by different structures in the process of non-cyclic photophosphorylation. So we simply want you to mention the structure involved, the molecules involved, but the mentioning should be systematic. One structure leads you to the other. All right, let me hear from people. I'm not seeing you people raising up your hands. Members, raise up your hand. Describe the role played by the different enzymes, meaning mention the first enzyme involved in DNA replication, in transcription. Actually, the first mark you'd get here would you mention that process by which mRNA is formed from DNA. So I would expect your introduction to say that the process by which mRNA is formed from DNA is transcription. You get your first mark, then you proceed. Yes, Amanda.
I'm just trying. So, this process is called transcription.
Okay.
the process, okay, the enzymes involved, we have helicase enzyme and RNA polymerase enzyme. The process starts by helicase enzyme. So we start by saying helicase enzyme binds on the DNA and catalyzes the unwinding and unzipping of the double strand, forming a fork.
Yes.
So on the transcribing strand at the promoter site, an enzyme called RNA polymerase binds and catalyzes the addition of RNA nucleotides following complementary base pairing. The RNA nucleotides are added until a stop codon is met. So after that, the mRNA breaks off and moves out of the nucleus through the nuclear pore into the cytoplasm. Yes, teacher, that's all I know.
Wow, that's wonderful. That is wonderful. The role played by the helicase enzyme, it catalyzes the breakdown of the hydrogen bonds that hold the nitrogenous bases together, hence unzipping the double-stranded DNA molecule. That's good. Then RNA polymerase enzyme. We have RNA polymerase. RNA polymerase enzyme has more than one role apart from assembly of the nucleotides. In the absence of the helicase enzyme, RNA polymerase enzyme can also catalyze the unwinding of the DNA double helix. If you read further about transcription, you will see the different types of RNA polymerase enzymes. We have RNA polymerase 1, RNA polymerase 2, 3, and so on. But 1, 2, 3, that simply describes the type of RNA that is being formed from DNA. For example, if it is RNA polymerase 1, RNA polymerase 1 catalyzes the formation of ribosomal RNA. That is rRNA from the nucleolus. Then RNA polymerase 2, formation of messenger RNA. RNA polymerase 3, that is formation of transfer RNA. So that's basically. So in case you are reading and you come across 1, 2, and 3, you should not get confused. 1, ribosomal. 2, messenger. 3, transfer.
Excuse me, sir.
You are excused, baby girl.
So, teacher, when they talk about enzymes,
Yes.
yeah, the roles played by different enzymes. Are we
Yes.
okay to also talk about DNA ligase?
Oh, that would be a correct enzyme, but for another process, that is, in case the question was, describe the role played by different enzymes during DNA replication. Then, ligase enzyme will be there, helicase, and so on. RNA polymerase, DNA polymerase, all those enzymes will be there. But for during protein synthesis, during transcription, RNA polymerase enzyme is the major enzyme that carries out all those three processes. Though sometimes DNA helicase enzyme also comes in, but majorly RNA polymerase is capable of doing all those three. I think let me show you an image here. Is it an image? An illustration of RNA polymerase. Then from that illustration, we can go ahead and give the roles of the enzyme. images. There's another image here. I think this is better. All right, there we are. Let me try to zoom in. Wow, this is better now. I think on your screen you can see the enzyme, that is RNA polymerase enzyme. That yellow one. I don't know whether it's yellow or cream, because I have two screens here. One screen is showing me yellow, another screen is showing me cream. So I'm even confused. Okay. One, we can see nucleotides being attracted to the transcription complex. So meaning the role of RNA polymerase here is to attract or assemble nucleotides, free nucleotides, to form the messenger RNA or the RNA transcript. Then in front of this, you can see a red circle. Do you see the red circle, members? Let me get for you a pen here. Where's the pen? Where's the pen? Pointer. Pointer. Okay, the point is there. So here you can see a red circle. Do you all see the red circle? Petronella, have you seen the red circle? This one here? Petronella was cooking and the teacher is calling her to come and speak. Petronella, are you cooking? Which food takes long to get ready? Beans.
Yes,
Okay,
teacher.
people are still. Finally, you are here. Were you washing clothes?
No, I was here but cooking also.
Yeah, I knew it. I knew it. So here we are seeing people are laughing. Betty and Bakita, stop laughing. Stop laughing. Stop dying with the laughter. Okay. So here we are seeing that unwinding of DNA, that red circle is carrying out. Unwinding of DNA. We shall eat beans in Feb. Okay, fine. We shall eat. Today she's cooking meat. So, RNA polymerase also catalyzes the unwinding of DNA. That's why you can see that red circle in front. Then this red circle behind, you can also see rewinding of DNA. So RNA polymerase enzyme can carry out three functions. One is to catalyze formation of the strand or the assembly of the free nucleotides, ribonucleotides to form the ribonucleotide strand or the messenger RNA transcript. Unwinding and also rewinding. I think that is all. Yes, Bakita, you have a question?
So I want you to rewind on the importance of RNA polymerase.
Okay, I projected this image because I wanted us to try to visualize the importance. So one, RNA polymerase here, we can see free nucleotides being attracted to the RNA polymerase. Do you see that? So the first role of RNA polymerase is to, okay, the role is to attract the free nucleotides or catalyze the assembly of free nucleotides to form a messenger RNA transcript. That structure of the molecule that is formed after transcription is the messenger RNA, or is the RNA transcript. Then secondly, I showed you these two red circles. The first red circle is showing unwinding of DNA. So meaning here, RNA polymerase enzyme is catalyzing the unwinding of DNA in that direction of polymerization. Then behind it, after formation of the messenger RNA transcript, behind it, it also has a mechanism that catalyzes the rewinding of the DNA. So after unwinding, the DNA has to rewind. And that is also the role played by the RNA polymerase enzyme. Unlike in transcription, where different enzymes carry out unwinding and rewinding, during messenger RNA formation, mRNA formation, or transcription, the process can be carried out even by a single enzyme, that is RNA polymerase. However, we mentioned earlier on that in some cases, helicase enzyme can come in. Okay. Wow. I've spoken a lot, and it's not always good for the teacher to speak a lot. I need to find out whether people were following or they want to give us more answers. If you're following, thumbs up. Betty is following. Amanda has given me a very big thumbs up. It is even bursting throughout the whole screen. Okay, that's good. So Makilena is lost. Catherine, I believe she's following. Just believe. Mwanje, you've been so quiet. Makilena is clapping. Catherine is laughing. Okay.
Yes, teacher.
Yes, talk to us.
Good afternoon, members. Master, I told you that the DNA topic, we had not yet covered it. So I'm trying to pay some good attention so that I can pick something.
Oh, that is wonderful. If I'm so fast, please, you need to ask me to slow down. Or if you feel that you'll follow up later, I'll upload the video on the channel. You can just go back there and try to go through it.
Okay, teacher.
Yeah, that's very good, Mwanje. Who else here? Bukenya, I saw you saying trying to give up. Is that sign for giving up or what? Yes, Bakita.
Teacher, I remember, okay, you said that RNA polymerase, it can also catalyze unwinding of DNA. Not so?
Yes.
So teacher, now what's confusing me is, does it performs the same function as the helicase enzyme also? Yeah?
It performs the role of the role that would have been performed by the helicase.
Bakita, see what your code again.
Betty, you are now.
Bakita, you're the one.
Chill me, I beg.
I know you're
confused. Is that my problem?
I'm going to write your names, those who are shouting.
Hey, frustrate yourself first.
I'm not joking. I write your names and they will clean you, no complaining.
You guys are serious when you're given a break, huh?
It's a talent.
Bakita chill, I will bring for you this.
Man,
By the way, Kathy, eh?
But
Kathy, you bring for us that's the what? Plum peanut.
yeah, wait, I'll bring for you this.
on top what Bakita is saying.
That one is for malnourished kids, and you're not malnourished.
Okay, I need it, you girl. Oh,
I'll try.
yeah,
But don't
good.
stop shouting. I'm going to write your names.
We are not shaken.
Okay. Who is teacher?
Kathy, me, I'm
Short
scared. Please write my name in capital letters. Eh?
I'm on the list. I tell you, fear canes.
Oh my God, on the way I'm met to you girls. You should wait for the day they'll cane me.
I tell with your sickness, they'll just give you enough.
Pardon?
Nothing.
Nae Kathy, if I were nearby, I'd give you nightmares today using a squeezer.
Macklin, stop lying.
I'm not lying. Hey, that color's gained weight.
Where Bakita should come and see me.
Now just
I've
because
kept
of
quiet.
network in our place.
My Bakita and blaming my God.
What are you doing?
My granny is taking care of me, so don't blame me for the network issue.
Just in Papa will give you in the chat.
I beg, slowly.
Guys, teacher is there.
Members, have you finished discussing?
Yes.
Yeah, you people, the moment the teacher disappears for even 1 second, what happens here is fire. Okay. What was I talking about, members? Remind me.
You are still differentiate. I was still asking you about RNA polymerase and helicase, the enzyme.
Yes.
Both of them literally perform the same function, teacher.
Yeah.
Yeah. So, teacher, now
So
okay, teacher, first try and break down that thing for me.
which thing?
Teacher, both of them perform the same functions, but when we are describing formation of mRNA from DNA, we talk about helicase alone, then we also talk about RNA polymerase alone.
Okay, while talking about transcription, the process of transcription, you can mention helicase and its role. However, in case you don't mention it, you are not penalized because it is not a must that the helicase enzyme must carry out the unwinding of DNA before transcription occurs. But it is a major enzyme that is required during DNA replication. While talking about replication or making another DNA copy from the pre-existing one, then it is a must you mention the DNA helicase, the role of the DNA helicase. But in case we are talking about transcription, you can mention about the role of helicase because it is the same role to carry out the unwinding. However, it is not a must that for every description about transcription, you must talk about helicase enzyme because RNA polymerase enzyme has a mechanism of actually carrying out most of the things by itself, unwinding by itself, attracting nucleotides, RNA nucleotides by itself, then rewinding by itself. Is that okay? Is that clear? Is that clear, members? Betty says it's clear. Bakita, the thumbs up are very big, meaning it is very, very. Let me just get here maybe another image that can help us understand it. Petronella, you want to go for lunch? I know your food is ready. It's about ready. I'm looking here for a simpler image here because these ones look complex to you people. Okay, I just want you to relate to DNA, rather RNA polymerase to a zip. Most of us have zips, not so? Or in case you have a zip on a cloth or a skirt or a bag or your bag has a zip. Those zips that we have on our bags work by unzipping and also zipping, meaning you can open the bag and also use the same zip to close the bag. So RNA polymerase also has mechanisms of zipping and unzipping, but the helicase enzyme only catalyzes the zipping. It zips and it goes away. No rezipping occurs. It catalyzes actually the unzipping, but it does not catalyze the rezipping. Hope that one is clear. I don't want to waste much time on it because Petronella was putting.
How are you doing? Like you even wanted to say it.
Unstable.
Even if my message is not saved, but that's okay.
Banyange.
Someone just reading what is listening to audio voices of WhatsApp and also making. I'm going to send that person in the waiting room, members, in case it prolongs. I'll send you in the waiting room and I lock the meeting. Let me just, members, give me a second. I switch on to the data of my phone. It seems this one here is creating more harm than good. Let me first disconnect it.
Hi guys.
Betty, don't make noise.
Don't want.
Let me play. Okay. It's connected to something else. Let me first disconnect here. All right, I think let me pray this one will be stable now. Hope you can hear me, members. You can hear me. Just do what? Okay, that's very good, Bakita. Let me first see what people are saying in the chat.
So
You
don't go to
always
that.
think
Don't go there.
I'm already
Yeah,
there. So
should not go to the chats.
do you have a perverted mind? Kathy, what's not happening? Kathy,
Then
people are talking about Kathy. Gloria, what is Kathy doing?
Kathy,
I'm in
prepare
a
for this. See,
CJ is there.
What is
Ashed.
okay, let's first come back to class because I want us to end by exactly. I don't want to lose any minute from 12:50. I really don't know how we shall make it, but let me try. Let's move on. Let's move on to the next question, members. Next part. That was 2. This is 3. Explain the process that results in the formation of an activated amino acid within the cytoplasm of a beta cell. This one I talked about it last time, members. Can someone come in and take us through very fast? Can someone come and take us through very fast? People are focusing in reading in the chat. Members, first come back from the chat. Don't go there. Seeing you are busy opening the chat. Yes, Catherine.
Okay, teacher, I'm going to try.
Please do try.
Yeah, the question says explain the process that results in the formation of an activated amino acid within the cytoplasm of the beta cell. So I'm starting like this. An amino acid binds to an ATP molecule to form amino acyl AMP, which is adenosine monophosphate. Then it binds to the 3' end of the transfer RNA under the catalytic effect of amino acyl tRNA synthetase.
So okay, that's very fine. So after forming an amino, rather after activity of the amino acyl tRNA synthetase, what would follow next, or the product that would be formed is an activated amino acid, or activated, or someone can call it amino acyl tRNA. Yeah, I think that is basically, and it is very clear and brief and precise to the point. There's nothing I should add on there. Because we had already discussed about this last time. I think those people who joined the last time can recall, are capable of recalling what we talked about. Hope that one is fine. Do you have anyone with a question? Let me screenshot Catherine's answers here. Let me just paste, screenshot your answers here, and I paste it here so that you see what you just talked about. Okay, let me come back to PowerPoint. Any person with a question? As I'm pasting here this girl's answers.
Teacher,
Yes,
I also want to try.
please do try.
So, okay, the process is called amino acylation. So the process starts with the amino acid activation. Here, the ATP molecule binds to the active site of the amino acyl tRNA synthetase enzyme, forming aminoacyl adenosine monophosphate with release of an inorganic phosphate. So this same enzyme binds on the tRNA molecule, and the activated amino acyl group is transferred from the aminoacyl adenosine monophosphate intermediate to the terminal of the tRNA molecule. Then the adenosine monophosphate is released, and an ester bond is formed between the amino acid and the tRNA molecule, forming an amino acyl tRNA molecule.
Okay, that's very good as well. It's very good. Your answers have been captured, and let's clap for them. Members, you are not clapping. People, you need to appreciate. Let me begin by clapping since you people have refused to clap. Okay, people are clapping. That's very nice. Those claps also go to Catherine. Yeah, that's very good. It's very, very, very good. Okay, let's proceed. I have nothing much to say. To me, that simply means that people were very attentive during that lesson. Let's move on today. Is it the last part or the second last part? Describe the role played by different organelles in the synthesis and secretion of insulin hormone. Wow, this is yet another question. When the examiner wants you to describe the role, he simply wants you to describe the process. But in the process, in case he has told you different organelles, in the process, mention an organelle and its function. Describe the role played by different organelles in the synthesis and secretion of insulin hormone. Mind you, the hormone, insulin hormone is a what? Is
It's a protein.
Yeah, it's a protein. I was just thinking about very many things, including I think my girlfriend maybe. But here we are, members. Don't worry. Can we
Teacher, please
can we have
concentrate.
someone again?
Oh God.
I'm
Teacher, no.
dying.
Set this in the papers. Wow, someone wants me to teach and refuse to set in papers. That's being unfair.
Teacher?
Yes. Who is speaking?
Mr. Wanyonyi,
We can hear you.
I wanted to ask, can you like send, I think, a PDF of answers? Because teacher, at some point, I don't know if I'm writing even the correct answers or I'm just writing what I'm hearing. But then teacher, send us the PDF of correct answers.
But you people have the correct answer. I'm just getting those answers from you people. Huh? I'm getting those answers from you.
Like a marking guide.
Okay, if you submitted the work, I'm going to include the marking guide in the system so that the system marks automatically, and I think it will send you the feedback in the marking guide. Hopefully, I'll try out and see whether it will work. Feed the answers in the system so that whoever attempts it marks you automatically there, and then it gives you marks, and it gives you feedback. I'm going to try and program that.
Okay, thank you, sir.
Okay, you're welcome. Bakita, you want to say something? Please say.
Yes, teacher, about this question, eh?
Mm.
So, teacher, for me, this question, it confused me so much. It reached a point where my answers, for me, I just listed them. Okay, I just put like mitochondria, then I put it's what its importance, it's used there like that, like that, like that. So that's how I did my work. So I don't know, even I don't know if it's correct.
Yeah, the good thing we are learning from today onwards, and that's why I gave out the assignment first. You try out, then you see where you present your work the way it is, the way you have understood it, then you bring, and we harmonize, we share and learn from each other. So whenever they ask you to describe the role or played by a given organelle or a given structure in a particular process, it means that this examiner wants you, indirectly, he wants you to describe that particular process. But in that process, those organelles must be mentioned and their roles must be highlighted in a systematic order. Is that okay? Someone would ask you to describe the role played by different structures of the kidney, nephron in excretion and osmoregulation. Should also be capable. Or describe the role played by different organs in maintaining a constant body pH. Describe the role played by different structures or molecules or organs in maintaining a constant maintaining a constant optimum pH of blood. So in other words, the examiner is looking for the process, but he just wants you to mention that particular organelle, that enzyme or organelle or structure and its function. As simple as that. If it is respiration, someone would ask you to describe the role played by different enzymes in anaerobic respiration, or describe the role played by different molecules in the electron transport chain. So they can just twist the question in any way. But at this, whenever you come across that from today onwards, describing the role or explaining the role played by a given organelle, they will always mention you maybe the organelles or different organelles, or sometimes they'll mention the organelle. Maybe describe the role played by RNA polymerase in DNA replication, rather RNA polymerase in transcription. So he simply wants, now there you'll have tied your hands. He wants you to focus on RNA polymerase alone. And stepwise, first unwinding, second attracting of the nucleotides and assembly, third rewinding, full stop. And you get your 4 or 5 marks. All right, let's come back here. The role played by different organelles in synthesis and secretion. So there are two questions in one. The first question is synthesis. The second question is secretion. So you should do merge the two, because you cannot secrete what you have not synthesized, meaning you should begin by synthesizing before you secrete. All right, let's go, let's go. One, let members here tell me. Do you have some people who have answers in the chat? Let me first visit the charts. Where are the charts? Yeah, okay. List the charts. 4 new charts. What are they saying? Don't set this in a paper, 17 minutes. I thought there were answers there. Yes, Bakita. Bakita is the only one giving us answers. Where are the rest? Members, let us
Sammy,
also
I'm just
try.
trying.
And that's why you'll become a better person. Because the moment you try, you give an answer that is partly correct and partly wrong, then you rectify it. Please go
Okay, teacher,
ahead.
I'm going to give one organelle and then its importance, yeah?
Yes.
So I have mitochondria, which provides ATP used in synthesis, packaging, and secretion.
Wow, so that's very wonderful. The mitochondria, it's where aerobic respiration takes place. Even the cytoplasm, we have respiration taking place. But having mitochondria, much ATP is secreted, is formed, is synthesized, which is used in various processes. That's just like she has mentioned. One is the activation of amino acids in the cytoplasm. You recall someone earlier on talked about it, that amino acids are activated by binding of an ATP molecule to an amino acid to form amino acyl, AMP, and so on. Then ATP still, the attraction of the free nucleotides to the strand, the growing strand of the mRNA also requires energy in form of ATP. So the mitochondria secretes energy, though that since it plays two roles, more than one role, you can mention it anywhere in your description. Wow. Continue. Is there any other person who has raised up their hand? Yes, Kathy.
Okay. Teacher, I also had the nucleus.
Good.
Nucleus, which is a site for transcription.
Wow. The nucleus
Then
is a site of transcription. It has enzymes that carry out transcription.
I continue.
Please, you can go ahead as we wait for others also to respond. Let me first see these people here. Kamori.
Okay, site for
Yes.
transcription, and we shall say a gene for insulin is transcribed into mRNA and sent to the cytoplasm.
Okay. Yeah, that's good. That's very good. So in the nucleus, it's where we have the transcription occurring. After transcription has occurred, there's RNA processing, mRNA processing. If you recall the other video we saw last time, during RNA processing, there is use of enzymes that carry out splicing. They remove the introns and they add a 5' cap and a poly A tail. But those two things, you don't need to stress your head thinking about them. You just need to keep life simple by just saying processing of the mRNA by removal of non-coding parts of insulin or introns.
Teacher, also ribosomes.
Yes, ribosomes. Man, ani augana. Achime MP sat. Ani augana achime MP. Kero ni chime MP. Come for slaps. Three. Yes, remember, Bakit is laughing. Why are you laughing?
So ribosomes are sites for protein synthesis. These move along the mRNA. As this occurs, a polypeptide chain is formed and this forms the insulin protein.
Wow, so ribosomes, they can be free in the cytoplasm or attached to the endoplasmic reticulum or forming what we call the rough endoplasmic reticulum, right? Yeah, and their role, the role of the ribosomes, she has already mentioned it, assembly of
Teacher,
Yes, Kathy.
what is the meaning of the insulin precursor? I got it from somewhere, but then I don't know its meaning.
Wow, I like the way you use AI to generate
No,
your answer.
so it's not AI.
Anyway. That is also
I got
good.
it
There's
from
no problem. Yes?
I got it from our friend and it was in Calibo, Isa.
Yeah, so the friend must have used the AI. Okay, but an insulin precursor. Um, what you have to understand in biology, the word precursor simply means either a structure or a compound that that acts as a raw material. Let me put raw material in quotes. That acts as a raw material for synthesis of another compound. For example, in case you like chapatis, making chapati, the precursor for chapatis is wheat flour. Do you relate that?
Mhm.
The precursor for making chapatis is wheat flour. So similarly, when you say an insulin precursor, that is the compound, or in simple terms, it is an inactive form of insulin. It's not yet activated. That is the insulin precursor.
Thank you,
Hope
teacher.
that you're welcome. Just like you have, in case we are talking about phytochromes in plants, during release of florigen hormone, sometimes we say florigen hormone is released from its precursors, meaning the raw materials or the inactive forms of florigen hormone. Then, in the same way, when we are talking about pepsin, the precursor for pepsin is pepsinogen. It is activated to form pepsin. The precursor for trypsin is trypsinogen, and so on, just like that. Members, I'm seeing it is almost time. Let's continue mentioning the roles of the organelles.
Teacher, we also have transport vesicles.
Okay.
Mr. Wanyonyi.
Yes, Amanda.
So these bud off from the rough endoplasmic reticulum to transport the folded insulin proteins into the Golgi apparatus.
Okay. Which part of the Golgi apparatus is it, the Cs or the transin?
Teacher, I won't lie, I really don't know. I've forgotten.
Oh, your voice is in slow motion. Okay.
Another one.
I said
Members, yes.
it's
We are
Kathy, not
listening.
me.
Yes.
Teacher, for me, my answer is done.
Okay, so the transport vesicles, they transport they transport the polypeptide chains that is containing an insulin precursor. The polypeptide chains from the rough endoplasmic, the pinch off from the rough endoplasmic reticulum, and transport the um, the protein to the Cs. Okay. Someone has not talked about rough endoplasmic reticulum. However, on a in case protein synthesis is occurring on a rough endoplasmic reticulum, we would see, you would visualize the growing polypeptide chain going through these people. Okay. Where?
Okay, they just sent us work on the group, eh? Sentaigwa Iseki. Wagwan.
Bakita. You'll get
I've not
what
seen
the
Bakita,
fuck?
but she's usually active when teacher is off, so don't blame her.
Please. They sent us an
Maclean.
activity.
Who is not?
What the fuck?
Now what, Kathy? I'm listening. Speak.
Yeah. What?
Okay, members. You see, being responsible is also very hard. People cry a lot, so they need attention. Yeah. What was I talking about even? I've forgotten. Members, remind me.
Endoplasmic reticulum.
Okay, fine. So the endoplasmic reticulum causes folding of the primary structure of the polypeptide chain that has been formed to form a secondary structure. Then this secondary structure is packaged into a transport vesicle and is transported to the cis end. And that is the cis end or the receiving face of the Golgi apparatus. Within the Golgi apparatus, what the Golgi apparatus does is to mark or label, carry out labeling and marking of that protein that has been received. The labeling is carried out by addition of a carbohydrate, specifically a glucose molecule, to form a glycoprotein. Or apart from labeling it that way, the Golgi apparatus will cause further folding from the secondary structure into the tertiary structure and also the quaternary structure. And this quaternary structure is then now enclosed in another transport vesicle, secretory vesicle, and pinches off from the trans end of the Golgi apparatus, then is transported to the cell membrane, then coalesces or fuses with the cell membrane, releasing insulin into the blood stream. Any question? If there is no question, you raise up your hand. If there is a question, you unmute and ask.
Teta, may I beg your pardon on the function of the endoplasmic reticulum from the point of labeling of chain received?
Come again.
I say that I beg your pardon on the function of the Golgi apparatus from marking and labeling of the protein received.
I think you have covered your microphone towards the end, but let me hope I have understood what you are asking.
I said that I'm begging your
Yes.
pardon from the point of marking and labeling of the protein received under the function of the Golgi apparatus.
Okay, so the goal. Do you have someone who is speaking as well? The Golgi apparatus I mentioned receives the packaged protein, the secondary structure, the slightly modified structure of the protein on the cis end via a transport vesicle. Then upon reception or upon receiving this protein here, the Golgi apparatus carries out further modification, and that is to mark and label this protein that has been synthesized. So during that process, the Golgi apparatus will catalyze, will have enzymes that catalyze addition of a carbohydrate group. In this case, it would be a glucose molecule. Addition of a glucose molecule onto the secondary structure or the quaternary structure of the polypeptide that has been formed, and that is to label it to form what we call a glycoprotein, a marked protein. So this glycoprotein, what the glycoprotein does, or what the Golgi apparatus does, it then packages this glycoprotein into vesicles which pinch off from the trans end of the Golgi apparatus. For me, I'm a visual teacher. I always like explaining while showing people what I'm talking about. Let me first get here the Golgi apparatus. I'm not going to open FA right now because it is very far, but let me access it via Google. This internet is loading, it's slow. Okay, let's just go right to the picture here. My face is so big and it is covering the picture. Let me first reduce. Let me block this. Oh, I hope here people can see now clearly. Let me just modify it by zooming in a little bit. Where has the image gone? Okay, there we are. Wow, this is wonderful. So from the nucleus, we can see the nucleus being in continuous with the rough endoplasmic reticulum. Then from the rough endoplasmic reticulum, we can see the transport vesicles. Let me first get this highlighter here, or pointer. Is it a pointer or a highlighter? Okay. So here we have a transport vessel from the rough endoplasmic reticulum, which binds to the cis, which fuses with the cis end of the Golgi apparatus. Then the Golgi complex or the apparatus carries out modification. Once modification has occurred, this Golgi complex on the trans end, this other side is the trans end. On the trans end, there is a pinching off of the vesicles, the transport vesicles on the trans end containing the insulin hormone that has already been labeled. Then this transport vessel fuses with the cell membrane to release insulin into the blood stream. As simple as that. I think that is clear. Members, clap for me if it is clear. If it is not clear, send an ugly face. Okay. If it is not clear, send an ugly face, a gloomy face. Okay, people are saying that they have understood. Let me hope it is not because of hunger, which is because we are about, it's lunchtime already, and people want to go and eat. So that is the last question. Okay, the very last one, members, I just want you to be very slightly patient. Try to pause the activity of the enzymes or the secretion of gastric juice. Just pause it for a while. I beg, I beg, I beg. Amanda, I beg that you pause. Kathy, I beg. Jerry, I beg. Explain the effect of reduced insulin production in Amina's body. So here we simply want the effects of, or the symptoms of diabetes in simple terms. Symptoms of diabetes. The effect of reduced insulin production in Amina's body. Symptoms of diabetes. At least people should be giving us one, and we go for lunch. Jerry, let me begin by you. Give us at least one symptom of diabetes. Jerry, unmute and talk to us. Jerry has left the room. Let me first check. Jerry, when I mentioned the name, he left. Okay, let me choose these people who are raising up their hand. I saw Kathy's hand up. Is that
Yes,
true,
teacher.
Kathy?
Yes.
Yeah.
And teacher, the answer is that increases glucose levels in blood, whereby there is reduced there is reduced rate of conversion of glucose to glycogen in the liver and the muscle cells. Thus,
yes.
thus levels of glucose in blood increase, causing diabetes mellitus.
Okay, causing diabetes mellitus. Sir, you are first. Is that comment just come now? I'm sorry if I'm fast. Or can you call someone for slaps? They were making noise. I just called them for slaps. Fatigue, constant hunger. Okay, the answers are here. Fatigue, thirst. Yeah, Catherine, your answer was nice. Though Mr. Sentai encourages us to always answer the question as we ask ourselves why until we can no longer say why. So in this case here, in case we have high level diabetes mellitus, so what exactly what exactly would result as a result of having diabetes mellitus? Someone would say glaucoma, a condition where there is increased glucose levels in the eye, especially the aqueous humor, which might result into blindness. A high concentration of glucose in blood. Constant hunger. Okay, that's good. Frequent urination. Yeah. Passing out urine with the glucose. So, which might result into UTIs, Moris, in case someone passes out urine with the glucose. The side effect, passing out of urine with glucose might not affect the person, but there is a side effect of passing out glucose with the urine with the glucose rather. One, since urine will contain glucose, it will make this person's body susceptible or vulnerable to UTIs. Remember, the fungi and the bacteria will always look for glucose, ready sources of glucose, and as a result, it would result into bacteria multiplying within the urethra of this individual. At the end of the day, in case it is not identified and treated early, it result into a UTI. Yeah, that's good. UTI. Reduced energy production, weakness. Someone talked about fatigue due to reduced energy production, or since there's low substrate concentration in this person's blood, we shall have reduced energy production. Then thirst. Thirst is as a result of the high osmotic pressure of blood. Increase in the osmotic pressure of blood that is passing around the hypothalamus stimulates the osmoreceptors of the hypothalamus, which trigger a reflex of feeling thirsty. And that's why those people who are diabetic always feel thirsty. Yeah. Then weakness or reduced weight, and so on. You can discuss more. It is already lunchtime, and I'm also feeling hungry. I think I've stopped thinking because of hunger. I need to go and eat, just like many of us here want to go and eat, unless Mr. Sentai has something to say. But
Mr. Wanan,
yes, please.
thank you so much
You're welcome, sir.
for adding a stone onto the struggle of these guys. Someone has been here in my inbox, and I'm like my pressure is seeing a person joining medical school, and then others joining is it called agriculture school? Then anything related to the reason why they are here. So you guys, it is good that you are here, and this is a sign that you deserve the success that you are going to get. So I'm just happy that you are learning, and because for us teachers, that's what we do. We are here to help people who wants to be helped. And Mr. Anil, thank you so much. There's nothing much I can say. I just want to tell those people that they should trust God, even the holiday, and protect themselves. That's what I can say. Have a good day. I'm off.
Wow, that's wonderful. Thank you so much, Mr. Sentai. Members, biology, Mr. Sentai, we shall meet Mr. Sentai tomorrow for biology. Then we shall also meet again on Thursday as our last day before Christmas. On Thursday, we shall be talking about the other scenario about cystic fibrosis. Make sure you go and attempt it. And when we come back on Thursday, we shall be discussing members. Yeah. Tomorrow, I think Mr. Yu should be there. Tomorrow, then Mr. Sentai. I wish you all the best. Nice lunch. Stay safe. Yeah. You are free to log out. If you don't log out, I'll log you out by force.
You're already out. Excuse me, teacher.
Yes,
How about you?
excuse.
The activity?
Which one?
The activity about we talked about cystic fibrosis.
I'm going to resend I sent it on Saturday, I sent it in form of a biology cake, but I'm going to resend it in the group. Are you in the group?
Yeah.
Send it in the group so that you get some time and access it.
Okay.
And Betty, you are still here. What are you waiting for? Betty, say something
Just know
as you
that
are logging out.
she is not around. She had already left. Now she doesn't know that the meeting is ended. She had just
Hey,
gone
yeah, I
to
get.
do other things. So
Wow.
we be getting people who have been just away.
Yes, so she doesn't want to say that she didn't attend the lesson.
She's there. She can even be up to evening.
Yeah, don't log out. Just be there.
Okay, man. We shall meet
We shall
later.
again to.
Um.
Okay, let me end.